What's up, distinguished boys and girls? We will discuss the cell division. The cell division is the process by which we get new cells from a mother cell. It's our body's way to make up new cells from older cells. I want you to understand that during cell division, the genetic material and the chromosomes will double. This happens in order to get equal distribution to the new formed cells. Before we actually get started, I want you to know that to master any cell division question, it depends on how much you study. It depends on how much you memorize. So really, this is the only way to master every question, because some questions can get really creative. So I want you to write it down on a paper, keep memorizing it, keep repeating it, and will it become easier. We have three types of cell division. We have amitosis, mitosis, and meiosis. Mitosis occur primarily in the somatic cells, and meiosis occur primarily in the reproductive cells. I will also discuss the gametogenesis, aka spermatogenesis, oogenesis, which is from the third chapter, because I think it is really complementary to meiosis. This is the main idea of meiosis. So I will discuss it. As soon as I finish the meiosis. Now let's start with amitosis. Amitosis is also called direct cell division or binary fission. So if you see those two terms, they mean amitosis. You can think about amitosis being a simple form of mitosis, because it's not as complex as mitosis. There's not much happening here. There's no spindle formation. There's no condensation of chromatin into chromosomes. Nothing much happening here, really. The book says, in this type of division, the cell divide without clear nuclear or cytoplasmic changes. The nucleus or nuclear matter and the cytoplasm constrict it and then split to form two cells, each of which contain part of the original nucleus or the nuclear matter and part of the original cytoplasm. So if they give you a question, give an example of an organism replicates by amitosis. It's bacteria or blue-green algae. Now, for the mitosis, we have nucleus division followed by cytokinesis. Cytokinesis is the division of the cytoplasm. Okay, so the division of the cytoplasm is called cytokinesis. This is how it looks like. So the cytoplasm is getting pinched, getting ready to get divided into new cells. Of course, the cytokinesis is different in the plant cell from the animal cell. We will also discuss this in detail, because in the plant cell we will have cell plate. We will discuss this. Now, as I said, the doubling of the chromosomes. You know that a human cell contains 46 chromosomes, but during cell division, it will get duplicated to 92 chromosomes, 46 going to cell and the 46 going to the other cell. The book mentions chromatids in this line instead of saying chromosomes. Now, one might argue this is correct, and one might argue this is incorrect. It's a little bit of both, but it's actually correct, and I'll explain you why. Now, in order to understand this, let's look at the structure of chromosome. We know that the chromosome is made up of sister chromatids, so this is chromatid number 1, and this is chromatid number 2. And here in the middle, we have something called the centromere. Now, the centromere is the structure that is responsible for attaching the two sister chromatids. I want you to understand that the centromere is also responsible for determining the number of chromosomes, meaning if I have one chromatid, I have only one chromosome, no matter the number of the chromatids. Now, let's say that I have this structure. Let's say that I have in the middle here, I have my centromere, and those lines are my chromatids. I have a lot of chromatids, okay, in one structure. This is technically, even though there is no such thing, but technically, this is still considered one chromosome. Why? Because I only have one centromere. But let's say that each chromatid will detach from this centromere, okay, and they will have their own centromere. They will become independent chromosomes by themselves. So this is exactly what is happening here. When the book mentions 92 chromatids, it's correct. At first, during the duplication, I still have only 46 chromosomes, with the sister chromatids making 92 chromatids, but those chromatids will detach from each other later in cell division during the anaphase, to be specific, and each sister chromatid will become an independent chromosome. Therefore, they will become 92 chromosomes by the end. I personally think if the book just mentions 92 chromosomes instead of chromatids, it will be more clear, because a lot of people get it confused here. And I can show you the image that is in the book that is showing it to showing the thing here. So we have, during the metaphase, we have the sister chromatids as they are with a one centromere, but during the anaphase, each sister chromatid will detach from each other into the opposite poles, and they become independent chromosomes by themselves. So by the end, we will have two nuclei, and each nucleus containing 46 chromosomes. Now, the mitosis goes through phases. But before we start with the first phase, we have something called the interphase. The interphase occurs before we actually proceed into the mitotic phases. How can you expect questions here? Now, you either are going to have straight question telling you to explain everything you know about the interphase of mitosis, or you can have it as a definition question, which is going to be very similar answer. But the beloved question really is, during which phase or stage does the following take place? They will just give you a fact that happens during a phase, and they will tell you during which phase or stage does the following take place, or does the following occur. Now, during the interphase, typically, the nucleus is relatively large compared to the nuclei of divided cells. Because here, we're going to have doubling of the DNA, doubling of the proteins, everything is getting doubled in the nucleus. So of course, the nucleus will bulge, will enlarge in order to fit. One thing that I want you to understand, the concept of nucleic acids. Now, the nucleic acids make up the genetic material, the DNA and RNA. DNA is made up of a nucleic acid called deoxyribonucleic acid, and the RNA is made up of ribonucleic acid. So when I say doubling of the DNA, I'm technically doubling my nucleic acid. So this here technically means the same thing a little bit, but in order to remember it anyways, in order to double my DNA, I have to double my nucleic acids. That's the basic thing that you have to know. And in order to, here we also have doubling of proteins taking place as well in the interphase. So doubling of the nucleic acids and the doubling of the proteins. So why do I need proteins? I want you to understand that in the structure of chromatin itself, chromatin is made up of the DNA and proteins. Okay, I don't know if it's mentioned in this book, but you have to understand, this is basic thing that you have to know, that the proteins are in the structure of chromosomes or the chromatins. So in order to double them, of course, I'm going to need to double my proteins. So this is only, I'm giving you the whole picture in order to remember this. We said that doubling of DNA will occur in this phase as well. And of course, the doubling of the centrosomes occurs in this phase. Now the centrosomes, we discussed this earlier. We said that they're the structure near the nucleus and they help in the movement of chromosomes during cell division. So if you have a question, during which phase or stage does the following take place? Doubling of the DNA, it's during the interphase. Doubling of the centrosomes, building large molecules of proteins, building large molecules of nucleic acids, all of them take place in the interphase. Now let's see the prophase. The prophase, in the prophase we have the condensation of chromatin into chromosomes. The sister chromatids will become visible. The chromosomes in themselves will become visible during the prophase. And the movement of centrosomes towards the opposite poles of the cell. And development of the aster, or the astral filaments. And between them we will have the development of the spindle filaments, as you can see in this image. So remember that the whole structure of the centrosome and the spindle filaments is in order to help in the movement of the chromosomes towards the poles. We will also have the nucleolus disappearance. Okay, so the disappearance of the nucleolus and the disappearance of the nuclear membrane or the nuclear envelope. And the sister chromatids that are becoming visible here are attached to the centromere. All of those steps are taking place in the prophase. Now, if they ask you a question, what is the location and function of the centromere? The location, of course, is in the chromosome, and the function, they connect the sister chromatids with each other. Very important. Also, do not forget, we have the nucleolus and the nuclear envelope disappear in the prophase. Now, what happens in the metaphase? In the metaphase, the chromosomes will get thicker and will shrink. And in the metaphase, the chromosomes will be located at the equator line of the cell, as you can see in this image. So let's see the metaphase. You can see that the chromosomes are aligned in the middle, like all of them are aligned very uniquely, very neatly, I would say. So this is what we call the equator line of the cell, okay? Remember, when you see this very neat, very unique thing, this is the metaphase. Okay, what else is happening here? The chromosomes attached to the spindle filaments fibers by the centromere. So, the chromosomes will attach to those spindle filaments only in the metaphase. We said that it will form in the prophase, but they will only attach the chromosomes in the metaphase, as you can see this in the image. This is also showing the difference between the spindle fibers and the aster. The aster is usually shorter than the spindle fibers. And we said that the spindle fibers are forming between the aster, between like from one to the other, okay? And the spindle fibers will attach to the chromosome by their centromere in the middle. So of course, during which phase or stage does the following take place, and the attachment of centromere to the spindle filaments? It's during the metaphase. Very important. During the anaphase, the sister chromatids will separate from each other, moving in the opposite directions of the cell, and as I said, when they separate from each other, they will become independent chromosomes by themselves. The mechanism of chromosome movement is not really understood, or is not fully understood. However, there are several explanations for it. This is a very important question. They will tell you, explain the mechanism of movement of the chromosomes towards the opposite direction of the cell. The answer is, you say that it is not fully understood. However, there are some explanations. First one is that the spindle filaments are thought to shrink when there is ATP, and they pull chromosomes toward the poles. So the spindle filaments will shrink in the presence of ATP, and they will just pull the chromosomes. The other explanations being spindle fibers form a path so that chromosomes slide on these threads toward the poles. So it's just forming it a way, and it will slide on it, and it will just move, okay? So during which phase or stage does the following take place? The separation of the sister chromatids? It's during the anaphase. And the movement of chromosomes, or movement of the sister chromatids toward the opposite directions of the cell, it's during the anaphase. The final phase here, being the telophase, is that when the chromosomes reach the opposite poles of the cell. So the chromosomes will return to chromatin fibers. So here, every process happened will just reverse in the telophase. So we said in the beginning that the chromatin will condense into the chromosomes. Here, they will just go back to the chromatin form. The nucleoli that disappeared, they will form again, and the nuclear membrane as well, that disappeared, it will reappear. The spindle filaments that appeared will disappear, and the nucleus division is followed by cytokinesis. Now, the cytokinesis is not really a part of telophase. Now, I don't know if this book really considers that a cytokinesis is part of telophase, because every time they mention cytokinesis, it's in the same paragraph of telophase. So I'm afraid that they consider cytokinesis as part of telophase. So that's why I want you to be careful if they have any question, like during which phase does the cytokinesis take place. So the book probably thinks or considers the cytokinesis taking place with the telophase. But the real thing that happening here, that the cytokinesis actually starts during the anaphase and ends by the end of telophase. I don't think you're going to get a question like this, but just a heads up, just be careful of any question, because I think the book thinks of cytokinesis being parts of the telophase. Of course, be careful of this question, like during which phase or stage does the following occur? the reformation of chromatin network, or let's say that the reformation from the chromosome to chromatin, it's during telophase. The end of nuclear division, telophase. The formation of nucleolus, telophase. Yes, formation of the nuclear membrane, the disappearance of the spindle filaments, and as I said, the cytokinesis, because I think the book considers cytokinesis as part of telophase, even though this is technically incorrect. Now, the cytokinesis, as I said, differs from the animal cell and the plant cell. Because in plant cell, we have something called the cell plate at the cell equator. As mentioned before, the cell plate is formed from the protoplasm. Okay, so it is formed from the protoplasm, and it grows from the center outward. Here, we say that the cytoplasm get pinched from the outside to the inside. So it's the opposite. Then each new cell begins forming its own cell from its respective side, ultimately resulting in two separating cells. So the cell plate will form the cell wall and the middle lamella of each new formed cell. But here, the cell cytoplasm will just get pinched from the outside to the inside and will develop new cells. Of course, the duration really depends on the events which occur in each phase. And the time for cell division varies according to the cell type itself, or the tissue type, and the age of the organism. The duration in human cells, and the prophase takes approximately 30 to 60 minutes, the metaphase 2 to 6 minutes, the anaphase 3 to 15 minutes, and the telophase is 30 to 60 minutes. So the metaphase is taken the less time, or the least time, and the prophase and the telophase taken the same time, approximately 30 to 60 minutes. And the division time of neuron during embryonic phases is also 30 minutes, but division of adult neurons is rare. Justify or explain, neuron division is rare in the adults. Very important question. Because adults, or the adult neurons, are specialized in a definitive way. Okay, so just keep in mind that the neurons do not go through cell division. They are constant, they are permanent cells.